disease killing the coral. And on the bottom right, you have, this is destructive fishing, there's dynamite fishing, but there's also overfishing that impacts reefs. So the concern is that we're going to have reefs that look like the left-hand side turning into reefs that look like the right-hand side. And that's the concern, because reefs on the left-hand side are highly productive. They do a lot of calcification. They grow. They provide all those ecosystem goods and services. We call them healthy. The reefs on the right don't. And so we call them not healthy. That's the concern. I do want to say, though, that I don't want to mislead you. Both of these pictures are from the same reef in New Caledonia. The one with all the coral is at the green circle, and the one without the coral is the red circle. Reefs, remember, are mosaics. They're patchworks of these different communities. So just because you find areas that are not coral doesn't mean the reef is actually not healthy. But I just wanted to make that that caveat, full disclosure there. So let's see if we can get to the next slide. Yes, we can. Well, we are concerned about reefs, and this looks like an eye chart that you might read at some, I don't know, not any eye doctor I've been to, but lots of small text. Don't worry about it. Don't worry about all the details of any of these plots. I'm going to make the point that's important here. Scientists are concerned about reefs, and managers, resource managers are concerned. The one thing that is measured in every study on reefs about how reefs respond to impacts, for example, the middle in the middle here, that's looking at reef response to a tropical cyclone over 30 years. And there's lots of other things, you know, temperature and all sorts of things. But the one thing that everybody measures, if you're able to see it, is how much coral is there. Coral cover, it's called. There's coral cover in percent. Coral cover, live coral cover, percentage cover, coral cover, percent cover. It's everywhere. Everybody from Bermuda all the way across the Atlantic, Pacific, Indian Oceans measures coral cover. This is a universal metric that reef scientists use as the first gauge of how the reefs are doing. There may be other things like biodiversity or how many fish there are, but everybody measures coral cover, and that's how reefs are compared, and that's how they're tracked over time. So coral cover, how much coral is there? Remember that, that's important, how much coral there is. All right, so how do you get that? Well, you get a lot of sunscreen, and you spend a lot of time on boats because you go diving. And these are all various methods. I'm not going to go through all the details of the methods, but what you'll note is with various cameras or tape measures, or I like the bottom one here in the middle, I call that shark bait. You get towed behind a boat for a while, and you write things down as you go. But everybody's diving. It's all scuba diving. And now I don't know if anyone in the room or online is a scuba diver, but if you are, you know that you can't go very far or very long. You're underwater for an hour maximum, and you can do that 2, maybe 3 times a day. And you're not going to swim very far. You're not going to cover a lot of reef. The only one that might cover a lot of reef is the bottom one, because you're being towed behind a boat. But that's actually not a quantitative survey. They get towed along for about 2 minutes, then they make a guess. Oh, I saw about 20% coral, and they write down 20%. So it's a very qualitative estimate. To get quantitative, hard quantitative numbers about how much coral there is, you have to spend time in a location making the detailed measurements, and you just cannot cover the area. So why is that an issue? Well, there are about 9,000 reef systems in the world, and they cover an area about 500,000 square kilometers spread across 200 million square kilometers of ocean. I did a back of the envelope calculation. I think that all of the reef surveys in the world ever maybe have covered into the hundreds of square kilometers. And I think that's being generous. That's 0.1% of the world's reef area. So that's not a lot. That's a very small sample. of where reefs are of the reef area of the world. And that's what we base our understanding on about reefs. I went further into this and I said, well, okay, let me start looking at data that I can find. I downloaded available data. I didn't ask anybody for their data. I just downloaded what I could. Most of it was from NOAA, the National Oceanic and Atmospheric Administration, and some other places, Australian Institute of Marine Sciences, some other places. And I took the in-water, these diving-based data, and I said, you know, they're not at the ecosystem scale. They're at the diver scale. An ecosystem is huge. A diver doesn't see the whole ecosystem. So what I did was I split the world into 1 kilometer grids, 1 kilometer cells. And in every 1 kilometer cell, I counted how many observations are there in that 1 kilometer cell. And that's plotted on the map here in the different colors. The purples are one observation per cell. The browns are 200 to 300. There, short answer, there's very few browns. Most of the survey areas have less than 10 observations. And I have it in a histogram here. This is in log scale, by the way. So this is, you know, to 2,000 some odd of the cells have less than 10 observations. Very few have more than 10. So we're trying to describe ecosystems with very, very sparse and unevenly spaced data. And I don't think that's representative of ecosystem scale. And I took those data on coral cover, and that's plotted on the left-hand side here. The y-axis is coral cover in percent. And I plotted that from all those data points in the previous slide against different environmental parameters. Things like the top row is various ways of looking at temperature stress, which is supposed to be important for reefs. This is the ocean acidification one. Then we have waves. Waves are important for reefs, then light, and then biodiversity and various local impacts. And I don't want to take a lot of time because this is a short seminar, it's supposed to be. But what we come out of this is what I've written on the right-hand side. Some of these factors, some of these environmental factors relate to coral cover as expected. Waves do. Coral cover goes down as there's more waves, for example. Coral cover goes up with more coral species. We sort of expect that. But a lot of them don't. So coral cover increases with coastal development threat. Coral cover increases with sedimentation threat. For example, coral cover increases with thermal stress. And there is no trend with ocean acidification. So this doesn't make sense. These data don't follow expected trends. That means either reef scientists don't know what we're talking about, which I don't think is true, or our data are inadequate. Low, as I said, low density, mismatched scales, or possibly the data are bad and we don't know what we're talking about. So let's find out. Wrote a proposal with JPL to NASA, part of the Earth Venture Suborbital 2 program for the Coral Reef Airborne Laboratory. I was the PI, partnered with JPL mission. The mission was managed out of Langley, and Paula Von Tempe, who's no longer there. She's now a dean at the University of Rhode Island. She was the program scientist at NASA headquarters. What was the idea here? The idea was, let's get data that are not diver-based. Let's get data at the ecosystem scale and see whether the trends still don't make any sense. That's the overarching question. So what do? We have 2 objectives. One, make all the measurements we can for high density, large area, and then do the analysis. Redo that analysis I showed you. And we went to the Great Barrier Reef, the Hawaiian Islands, the Mariana Islands, Palau, and we got some bonus reef in Florida. What we did was we used the technology called imaging spectrometry. And we put a very nice camera, I'll show you that in a minute, in the belly of a Gulfstream 4 jet. and flew it over reefs and used the spectral signatures of coral and algae and sand to identify what's there, to say how much coral is there. And so that's what we did. I'll explain it a little bit more as we go. We had wonderful partners. I don't want to give them any short shrift. Wonderful partners across the US and international made this all happen. So the instrument, for those of you who are engineering inclined, I'm not so much. So this is a bit of a canned slide. The top right is the CAD drawing of the Portable Remote Imaging Spectrometer, or PRISM. PRISM on the bottom right here is in its vacuum chamber, gets chilled to very low temperatures to maintain a stable data collection. We have some requirements. What this thing does is it measures a spectrum at every pixel in an image. So a plane flies along a reef or wherever, and it scans the Earth's surface. And at every pixel, it gets a full spectrum, not just red, green, blue colors that your phone gets when you take a selfie. It gets a full spectrum. It gets all the colors, and it differentiates them all out. And so that lets us tease out a lot of things about the reef. And so this is the technical basis. The Portable Remote Imaging Spectrometer, this was designed for coastal observations specifically. And without going into the performance, it's excellent. This thing does the best job of any instrument that I've ever used for this sort of thing. And you see on the bottom left, our JPL people looking in the hole that was put in the bottom of this Gulfstream 4 jet. Interesting note that this was a luxury, business jet with nice wood paneling and leather seats and all that. And they cut a hole in the bottom. And the moment they cut a hole in the bottom, the plane lost its FAA airworthiness certificate, and it became a NASA experimental aircraft. So I wasn't allowed to fly in it, which made me sad. But it worked perfectly for us. There's the airplane in a hangar in Cairns, Australia, which is in northern Queensland, just to show you what the plane looked like. Again, nice jet, flew high, about 28,000 feet, and very fast, about 400 miles an hour. And we got a lot of data. All right, I'm going to switch gears before I get back into the remote sensing. I just want to show you we did validation. We're doing all this stuff from the air. How do you know it's right? Are we making it up? Well, no, we're not making it up, but we have data to demonstrate that. So we measured this the reflectance. This is the light leaving the water. This thing floating here, this black thing measures light leaving the water. This white thing measures light coming into the water, and we can use that to test part of our algorithm out. That's the airplane measures light at 28,000 feet. Well, the reef is underwater, so we need to do some corrections for all the air that's in between, and this lets us check that to make sure it's right. Just some pretty things. This is how it works. I'm not going to go into the details here. It just says that we made a lot of observations. That's a picture of it. This is a picture of it underwater, and some fun things. These are what the data look like. These are spectra. On the x-axis of this is wavelength. 400 to 750 nanometers. 400 to 700 is visible light, with the violet being on the left and red being on the right. 7 to 750 is infrared. And this is various bottom types that we're looking at. The top is sand, and you can see the pictures, and those are a bunch of sand spectra. And then we have a bunch of algae on the right. Those are algae spectra, and then we have coral. Those are coral spectra. So that's just to show the data that we got. And we can use those to make sure that our algorithms are working right. We also deployed this beast, and it is a heavy beast to pull in a boat. That measures water optical properties. That's how clear the water is. Is there a lot of phytoplankton in the water? A lot of floating single cell algae? Is there a lot of sediment that's stirred up? That's what we used this instrument for. Here it is on the right-hand side, hanging over the side of the boat on top of a reef with lots of coral. Very pretty picture. Again, the left-hand side, not important for this seminar, but just we did a lot with these data. Okay, there is yours truly diving with a spectrometer. I'm measuring the color of the sea floor, making a lot of measurements. I happen to be pointing the spectrometer at algae at the moment. I had just done it on coral, swimming back and forth, measuring the spectra underneath the water on the reef surface. So we have light, we have spectra from the airplane, we have spectra at the water surface, and we have spectra at the reef surface. So at 3 different levels, and we can use these data to make sure that what we're doing is correct. Here are some examples of some sites, some data. Not important. The left-hand side is mostly algae and sediment. The right-hand side is mostly coral, and so we can use that. Just the details don't matter. Not for this talk. We did a fun thing to say how much coral is there. We did underwater surveys, and we used a process called structure for motion or photomosaicing. We have divers put down scale bars in a pattern like this on the bottom left. You can see one diver putting a scale bar down there on this coral, and then they swim back and forth, taking 1,000 photos, putting it through software on a computer, which stitches the photos together. And actually, on the right-hand side, all those little blue rectangles tell us where each of the photos was when it was taken, stitches it together and lets us make a mosaic of the reef, which looks something like on the top left-hand side. This square mosaic on the top left is about 10 meters across. You couldn't take a single photo 10 meters across. But we make a mosaic, you can, and then you have these little red squares. You can just to show you that you can zoom in. You can really see detail on this top square here in the middle. Those little dots, that's a coral, and those little dots are about 1 millimeter across. Really small. You get really high resolution, so you can really see what's there. And we can use these things, and we overlay a whole bunch of points, and we count how many points are coral, how many are different kinds of coral, how many different kinds of algae, and we get the data that's on the bottom here, on the bottom left. And that's the benthic cover that we're talking about. Back to benthic cover, that's how much coral there is. This site happened to have about 85% coral. So here's an example on Heron Reef in the Great Barrier Reef, where we show the color-coded the sites that we went to based on how much coral, algae, or sand. You can see this triangle here, this ternary plot. If it's all coral, then the dot will be red. If it's all sand, the dot will be blue. If it's all algae, the dot will be green. Then, of course, you have mixtures. So this has a lot of coral. This green dot has a lot of algae. This pink dot is a mixture of coral and sand, and so on. So we have these data in a lot of places in Australia, Hawaii, Guam, and Palau. We also did metabolism of the reef. That's measuring the photosynthesis of the reef, and this is a whole project unto itself. In fact, I have 3 research experience for undergraduate interns here with me this fall for the next 3 months, and we are deploying this very set of instruments on the reef here in Bermuda. Just got back actually 2 hours ago from doing that, so I had a nice dive this afternoon. But this instrument, without getting into details, measures the photosynthesis of the surrounding reef. There's another way we do it, which measures upstream and downstream with the water flow. We put other instruments out that also measures the photosynthesis of the reef, and this one also lets us get the calcification rate. So that's how fast the reef is building its limestone skeleton. So we did that. Okay, that's all the validation. Let's get back to the remote sensing. So here are all of the flight lines that we had across Australia in September, October 2016. Then we went to Hawaii in February, March 2017. We went to the Mariana Islands in April of 2017, and Palau in May of 2017, and then finally Florida in May of 2017. And all those green rectangles are the outlines of the flight lines. All of these data that I'm talking about so far and going forward are publicly available. And if you don't have time to write it down here, in fact, this is out of date, actually. There's a better place to get it at if you go to earthdata.nasa.gov, earthdata.nasa.gov, you can search coral, the Coral Reef Airborne Laboratory, and you can find all of our data. So earthdata.nasa.gov. We covered a total of about 75,000 square kilometers of the Earth's surface. About 10,000 of that was reef surface area. So we got about 2% of the world's reef, which is far more than ever has been before. So that's pretty good. And we did in-water validation, like I said, everywhere except Florida. And those in-water data are also publicly available. So if you want all of those fun data, you can have them. And that's at, this website is correct, cbas.gsfc.nasa.gov/experiment/coral. And if you have, I'll provide that to you later if you want, no problem. Oh, Earth data. Huh. I should have known what the next slide was. Here's the Earth data webpage. You can go to Earth data, you can search coral, and you can find our flight lines. You can each flight line is individual. It's in net CDF format. So please download and use and enjoy. One thing we do, and here's just some details here and there. The airplane flies at 28,000 feet. We have to correct for the atmosphere. The atmosphere actually gets in the way because there are molecules of gas, there are particles, aerosols, all kinds of things in the atmosphere that actually make the sky blue. But that gets in the way when we're looking down at the Earth. So we have to correct it. On the left-hand side is an actual corrected image. We left the clouds in, but everything else is corrected. What we see, there's a problem here that there's glint, and that one side of the scene doesn't have glint. The other side has the sunlight is bouncing off the water surface back at the sensor. And we correct for it, and that's on the right-hand side. Now you see a nice, smooth picture, and you can actually see through the water a lot better because this glint from the sea surface is gone. The right-hand side just shows the impact of correcting for glint on the data here, our spectra on the right-hand side, and glint just sort of lowers it like that. I wish I had 4 hours to go through the algorithm, but I don't. We're using a simplified shallow water reflectance model. I know it's a lot, it's a mouthful, and it's going to get complicated, and I apologize. But basically, the model uses 4 parameters. There's scattering in the water. That's how the water light bounces off water and particles and comes backwards. This is the attenuation. That's how fast light disappears as it passes through the water. There's the water depth, which is H. That's just how deep the water is. And then there's how much the bottom reflects light. So all of these things put together in that equation give us the color of the water at the water surface. And so we use that to, we sort of go backwards. This is what we're measuring, the left-hand side of the equation. We're measuring the left-hand side, and we use some fun numerical methods to estimate the backscattering, the attenuation, the water depth, and very importantly, the bottom reflectance, because that tells us whether it's coral or algae or sand. Man, as I said, I could talk for hours. We parameterize the model. We have some good ideas of what K should be. That's the top row here. Won't go into detail. We have some good ideas of what the backscattering should be. That's the bottom row here. So we can model K, and we can model BB very well. We can do that very well. If you compare the right-hand side to the left-hand side, we can do that. So we can use these in our fun numerical approaches to figure these out. Depth is just depth, and bottom reflectance is modeled differently. I don't have anything to show you on that, thankfully, because that would just go on forever. But these are some example retrievals of the K that I was talking about. It's hard to see on the left-hand side, but we're in water measurements that are these X's. I don't know if you can see them here. You can see them better in the middle one. The X's are the in-water measurements, and the circles are what we retrieve from our algorithm. And so we're retrieving pretty good estimates of the water clarity. That's what the K is. It's water clarity. And we're getting good estimates using our algorithm. This is showing the retrieval of depth. On the left-hand side is a scene of a bay in Hawaii. In the middle of these images is LIDAR data, LIDAR bathymetry. That's using lasers to find out how deep the water is. That was done in 2013 for most of the bay. On the right-hand side is the result of our algorithm. So this is the retrieval from our algorithm, and we compare it to the LIDAR data, to the laser data, and that's on the right-hand side. The x-axis is the laser, the LIDAR data. The y-axis is our retrievals, and we get a pretty good retrieval. It follows the one-to-one line, so we're doing pretty good. This is the retrieval of benthic cover. This is what we want for Heron Reef, Australia. We have a scale bar. I don't have anything telling what the colors are, so I have to tell you. It's the same as before. The reds are mostly coral. The blues are mostly sand. The greens are mostly algae. And when you have colors in between, like purple or orange or yellow, that means there's mixtures. And the accuracy of our retrievals it approaches that of in-water observations. We're getting really accurate results. I'm very happy about this. It took a while to get here. I worked on the algorithm, revising it for several years. And you're the first people to see this result. So I don't know if you feel special or not, but you're the first. This is just new this summer. And finally, we're ready to actually get these out. I'm very excited to get this in a publication. This is the algorithm for photosynthesis, which is primary productivity. Complicated stuff. Won't go through it, but I just want to demonstrate that we've actually thought about it, put a lot of effort into it. It's a model. It uses data from the imagery that we get, and it uses other data sources put together to make estimates of photosynthesis. That's on the right-hand side there. So put it all together. Here is an example data set. On the left-hand side is just sort of your true color Google Earth type picture. The next picture over is, this is the water clarity. This is what I was talking about, how this K parameter, the water clarity, where browns are higher, turbidity, less clear water, blues are more clear water. Then we have water depth, which I showed you already. The bottom, reflectance. This is what we use to estimate the benthic probabilities of coral, algae, and sand. That's here, and this one here with the triangle again, with the corals red, algae's green, sand is blue. So that we estimate the benthic probabilities. We're also estimating primary production and calcification from the primary production. So far, we're only really working on the coral, algae, sand part. The far right-hand side, we're going to come back to those in the coming year or so. I did not show this earlier. This is just to show you that we have data from other sources, not from coral, but we have data about light and thermal stress and ocean acidification and waves and all kinds of things that we have that we're going to compare our airborne data, the products that I'm showing you, against. These are the environmental variables, the environmental parameters that we want to use. So what we do is we take the coral data. On the left-hand side are 12 flight lines from the Torres Strait. The Torres Strait is the bit of water that's between the northern tip of Australia and the southern tip of New Guinea. So Australia is in the south and New Guinea is in the north, and this is the Torres Strait. And that's the flight lines with PRISM. There's 12 of them. In the middle, we have our probabilities of coral, algae, and sand. And these are 8-meter pixels. The pixel size is 8 meters, actually 7.9, but we'll just call it 8. At 8-meter pixels, I take these 8-meter pixels and I figure out which ones are mostly coral, which ones are mostly sand, whatever, and I add them up. I add up all the pixels in a 1 kilometer square. And then I use that to get how many pixels were coral out of that 1 kilometer square. And that's how we get coral cover. So we surveyed the whole kilometer square of each of these cells with a whole bunch of 8-meter pixels. And we got the benthic cover for each of those squares. So there's a whole bunch of squares there. So this is what the data might ultimately look like. On the left-hand side is the coral cover from the previous slide. We have the photosynthesis on the next one. We have ocean acidification variable. We have thermal stress variable. We have waves, and we have various local threats: coastal development, marine pollution, overfishing, sedimentation. And you can see that for each square of coral cover, we also have a square of each of the other parameters. And so we can use that to do analyses. And this is provisional with a bit older data. I'm still working on doing the newer data, but still unexpected trends. We still don't have the answers we were looking for. So, what do we do here? Coral data do not follow the expected trends with respect to environmental factors. So one thing I take from this is that we can't just look at coral cover against thermal stress, because thermal stress isn't the only thing that's driving what a coral reef looks like or what it's doing. Waves also play a role. And you know what? So does diversity, and so does ocean acidification, and so does light. All of these things are acting all at once. So it's not coral cover against temperature. It's against everything, right? And often in reef science, it's presented very simplistically that it is one thing or another, but it's not. It's everything. But what coral does allow here, I want to point out that for the first time, we can do this sort of quantitative modeling of the complex interactions between reef condition, that's how much coral there is, and the environment this is at the ecosystem scale. No one's ever been able to do the ecosystem scale before. So this is the big deal, is that we're looking at the whole ecosystem. We're not missing the forest for the trees, not missing the reef for the corals, right? We're seeing the ecosystem, not the corals. We're counting coral, but it's at the ecosystem scale. One thing that we can do, though, is, as I said, it's not one thing or another, it's all of them. So we shouldn't be using this simple XY plots. We should be doing fun multivariate analyses. So that's what I did, trying to say, well, how do all of these things together relate to coral cover, right? So I put them together, and on the left-hand side, I went back to that in-water data set I talked about at the very beginning, and I did the best possible model I could do, just statistical model, and I get this R squared of 0.38. That means that this is between the true coral cover and the predicted coral cover from this model that I made. That means that the model I made does not do a good job. It's only accurate about 38%, right? There's a lot more that's not being described, that the model's missing a lot. But when we use the coral remote sensing data set, we're getting an R squared of 0.64. That means the statistical model that I built with the remote sensing data set is doing a much better job. So relating coral cover to the environment really needs the ecosystem scale. This is my takeaway. We really need to be looking not at the corals as organisms, as little tiny things, but at a kilometer. That's how we have to be looking at the reef. With that, I do believe that is my last slide. I got to thank the science team. That's at JPL on top, and the bottom is the last field photo from Palau in May of 2017. Everyone here made it happen. It was great. I'm sorry I went a little over, but I really appreciate you paying attention. I hope I didn't overwhelm you with too much information. I'm really happy to take any questions you might have. Thank you.
Thank you, Eric.
Thank you, Eric. That was really cool. Any questions from students in the room before I move on to not students in the room? In the back.
And we have a microphone for you, so if you just hang on for just a second, we'll get the microphone to you.
First of all, thank you for a great talk. So forgive me if I misunderstood, but just to clarify, was this the first time that spectroscopy has been used to study coral reefs before?
No, not the first time it's been used to study coral reefs. It's been demonstrated actually for the past 25 years. This is the first time we did it in such a large applied way that we actually went out and did it across the Pacific Ocean. It hasn't been done at that scale before. So it's been done here and there. It's been demonstrated. Hey, look, we can do it. And finally, in fact, almost 10 years ago now, we started convincing NASA that yes, we can do it. Give us the money to go actually do it. And that's how it came about.
Gotcha. So my main question was, because I never heard about spectroscopy through water personally, just because that's not what I study. But I was wondering, did differences in like water quality or composition or differences in the underlying sediment greatly affect the results of the spectroscopy? And or do you have to like correct for those factors, or did they not play a role?
Well, so the sediment is part of the retrieval. We want to know that. So we hope it affects the results because we want to measure it. As far as the water is concerned, it does impact things, but I didn't get into the algorithms too much because that's a whole other thing. But the algorithm corrects for that up to a point. Once the water gets too cloudy, you can't do anything. So, but up to a point, we can correct for it. So that's part of the algorithm.
Great, thank you.
Right here in the second row.
Wow, she's waiting for a microphone. I have one online here. So how is sedimentation threat assessed? And do tropical storms and floods increase the sedimentation threat?
I missed the first part. I got the second part. How is sedimentation threat? Oh, so okay, I see. I opened the chat window so I can see that. Okay. So I got the a sedimentation threat and all the local threats I took from a resource called Reefs at Risk version 2. That's put out by the World Resources Institute, I believe, and they put out a GIS data set where they spent the millions of dollars to assess sedimentation threat. I just used their data. As for tropical storms and floods, so what happens when a cyclone comes through is it resuspends a whole lot of sediment, and it's a really sharp, acute event. It happens really fast, and a lot of sediment gets put up in the water. If there are currents, the sediment gets washed away. If there aren't a lot of currents, the sediment will settle back down and may get on top of some things or not. It doesn't really bury anything, in my experience. It doesn't seem to really increase the threat. Floods, now as sea level rises, we're going to have more flooding, and that's going to get more terrigenous, more land-based sediment out on the water. I don't know what that's going to do, but that's a good question. I don't have an answer for that.
Hello, can you hear me? Thank you for your talk.
Yes, I can hear you.
So I understood that coral cover versus temperature is not a meaningful metric, but if you could include as many factors as possible, so acidification, pollution, thermal stress, etc., in your model, and then compare it with remote data, is it possible to confidently assess the specific impact of each factor on the coral reef, and how confident would you be that this is a global trend among all coral reefs?
That's a great question. I don't think that temperature is meaningless. I think that the results that we got from the in-water data are flawed because of the data, and I'm going to withhold judgment from the coral results because those are still really in progress at the moment, to be honest. But you're absolutely right. So what you can do with a multivariate approach, you can put all the variables in and build your statistical model. And you can see how well does the model overall perform. And then you can test how each of the variables impacts the model results. You can do a partial dependence analysis, and you can see which ones are more important. And so, yes, you can do that. And that is something I'm actually in the process of doing. I just don't have the results to show you.
We have another online. Can you see the one from Alexander? I'll read it for the audience here. Coral's required spectral range of 400 to 800 nanometers seems limited to visible light. Are there potential benefits to expanding to infrared and ultraviolet?
That's a great question. So, it was a little bit, we left some information off of that. That the requirement was, so that requirement is for the algorithm, the through water algorithm. So to correct for the water, to estimate the depth and get the bottom cover, we need 400 to 800. That's what we need. We could use more to some extent. To do the atmosphere part, we don't need the visible, we do need the infrared. So the overall requirements were basically 400 to 1,000 nanometers. We used the infrared for the atmosphere. So that is definitely useful. Water very strongly absorbs infrared light. So infrared is not useful for the water part of it and the actual reef part of it. It's useful for the atmosphere. Ultraviolet is a very interesting question. There's not a lot of work on ultraviolet with reefs. And also, an issue though is that calibrating the sensor at ultraviolet wavelengths, even the far blue, calibrating it is problematic to get a light source, to get a good calibration. In fact, we cut off the data at 430 nanometers because below 430 it was too noisy.
Any other questions from our audience?
So one of your models said it was for shallow water correction, and it looked like
Yes.
all of our sites are fairly coastal or shallow. So what depth are you limited to, and would it be useful at all to look at corals that are in deeper seawater?
Well, that's a great question. So we're using light, and so we're basically limited to how deep can we go that the light reaches the bottom, gets back through the water, and gets all the way up to the sensor. That's not too deep. So with some satellites and some, you can see pictures just like satellite, like in Google Earth, you can see down as deep as 30 meters, 40 meters. You can see that there's reef there. Those satellites don't do anything for the actual telling you what it is. With our project, so the threshold, the minimum is 5 meters. We need at least 5 meters depth. And that's useful because most reefs, well, virtually all reefs have what's called a bimodal depth distribution. They're either shallow or deep. There's not a lot of in between. And so, and also, so it's shallow 5 meters or less, or 5 meters or shallower. So we get a lot of reef area at 5 meters. Also, that's where all the activity is. That's where all the light is for the growth, the photosynthesis. So 5 meters would give us a huge important data set. Although that being said, with my new reprocessing that I'm working on this summer, I'm pushing it to 10 meters. So that's about 30 feet deep. I'm not going to push it beyond that. 30 feet is more than enough to give us all the reef that we need. Now, the deep reefs that you're talking about, corals do grow, for example, off Hawaii, down to 100, 120 meters. We're not going to see them from the air. In fact, you need a submarine to see them. So, but they're also not building reefs. They're just corals growing there. We're focusing on the classic sort of shallow water reef. Because that's what we can see, but that's also sort of what's important for all these ecosystem goods and services.
Over here.
I'm curious about how your, let me back up. Greg Asner from here in CC and his group are doing coral reef related monitoring, but I don't know what's similar, what's different, what's overlap with what you guys are doing?
That's a good question. I cannot speak for Greg at the moment. When we started, when Coral was funded, Greg approached me and JPL about collaborating because he was doing a lot of terrestrial work, but he was, he lived part of the year in Hawaii. And he had his airplane there with his sensor, and he said there was a postdoc who was doing a small reef project, and he was going to fly over the reef for her, and he asked if we could collaborate, and we said of course. So we collaborated, and I think you got to ask Greg this. I think that really whet his appetite and got him going in the reef direction. So he got the data, we worked on the data. There's a publication that came out of it, and I don't know exactly where he's at. We sort of diverged. I was so focused on doing the mission, and he was focused on his stuff. I think at that time he was moving from where he was to ASU and things. You have to ask him. I don't know. I'm just,
You need to talk to our own colleague.
I'm sorry?
I will talk to my own colleague about this.
Yeah. But so I don't know where he's at in terms of the algorithm and what he's doing with it. I don't know where he's flown. I know he's flown Hawaii. I don't know if if he's done like the whole Pacific kind of deal, like, well, not the whole Pacific, but across sort of thing that we were doing with Coral. So yeah, I don't know.
All right. Well, we're out of time. So if we could thank Eric again for a great talk.
Thank you very much.
And thank you so much for giving a talk at 8:30 at night.
My pleasure.
All right. Bye-bye.
Thanks, everybody.